In the center of the DNA double helix, purines and pyrimidines are hydrogen bonded together. This represents one such pairing, guanine, a purine, hydrogen bonded to cytosine, a pyrimidine, through 3 hydrogen bonds. The hydrogen bonds are illustrated in the center of the cytosine-guanine pair. Cytosine, the pyrimidine, is made up of a single ring of nitrogen and carbons, while guanine is made up of 2 rings. This picture illustrates the other purine-pyrimidine pair that can be found hydrogen bonded together. You will notice when adenine on the left, the purine, and thymine on the right, the pyrimidine, are hydrogen bonded together, there are only 2 hydrogen bonds versus 3 for a CG pair. You will again notice the hydrogen bonds in the center of the bonded pair, the pyrimidine, thymine, made up of 1 ring, and the purine, adenine, made up of 2 rings. To distinguish which end is the 5' or the 3' end, you must count the carbons in the deoxyribose sugar. Carbon number 1 is always the carbon attached to the nitrogenous base, and we then count away from the oxygen around the ring. In the center of this model, you can see a CG pair with the 3 hydrogen bonds holding the nucleotides together. Here's the cytosine, the pyrimidine, and here is the guanine, the purine. Down below, we have an AT base pair where we have 2 hydrogen bonds holding the purine adenine onto the pyrimidine thymine. The same pattern applies to the large model. Carbon 2, 3, 4, and 5 on this sugar, 1 attached to the base, 2, 3, 4, and 5 on this sugar. Now here we have a base pair with 2 hydrogen bonds. Can you identify thymine as this pyrimidine? Above, we have a CG base pair. The single ring shown here would be cytosine, and the 2-ring purine would be guanine. On the side of the model, we have a phosphodiester linkage attaching the sugar of one nucleotide to the phosphate group of the next. The purple molecule is phosphorus as part of this linkage. Can you recognize what base pair this is with the 2 hydrogen bonds? Now try and see how many of the previously named structures you can identify on the small DNA model as it rotates.